Do you all guys understand how they work? I mean, what's the difference between them?
Could you please explain it one more time?
Okay, so you have a sample. It's placed on the slide. And this sample is your cell. So you have a nucleus here, and here is your objective. And let's say it has some set of lenses to enlarge. And also we have a source of energy. So this is light source. And we assume that the light source is in some distance. So we have parallel light rays that are coming through this sample. So when they pass it, some of them are being scattered. Some of them come through, but in a very low powers. And others just go through. So according to these light beams, we are having the enlargement, and this enlargement will be seen like some kind of light structure and dark structure because of this scattering. And since we are using light as a source of illumination, as a source of rays that being scattered or passed through, we call it light microscopy. Okay, so then we switch to electron microscopy. In this case, we have a sample that is placed on the stage. And we have the source of accelerated electrons. And this source creates the beams of electrons, not the light, but electrons. And since their wavelength is much shorter than the one of light, we can overcome the limit of the visible light. So these beams, they are also kind of come to the sample, and some of them, because of the structures in this sample, they are being scattered or reflected. And we have a set of detectors that kind of catch these reflected or scattered electrons. And if they catch it, they say, okay, I have one electron scattered from this place in this direction under this angle. And on the computer, well, this detector, for example, sends this information to computer. And on computer, the processor starts computing the shape of the object, because of course this detector caught not one but many electrons. So all this information goes to your computer, and it's computing the shape of the object, and it starts building it. And because we have detectors that are placed in a 3D space, so we have several detectors, and they catch all these electrons, they can build a 3D structure. So it says like we have a cell with a nucleus here, and all the invaginations and protrusions of the cytosol, etc. And it detects everything. And because we have higher frequency here, which means lower wavelengths of electrons, it can catch very, very small differences in structure. That's why we have so much detailed images of electron microscopy. So as you see, we have different composition of the microscopes. And first of all, we have different source of illumination. In one case, we use accelerated electrons. Okay, so first of all, they are different in sources. One uses electrons, another uses light. Second, in the light microscopy, we use physical lenses made of glass. So it's glass lenses. And in electron microscopy, we use curved magnetic field. So we have two sources of magnetic field. And passing through this magnetic field, these electron beams, they are being bent. And it almost looks like the path of the light that goes through this lens, for example. And this is the difference. So the question about the oil in refraction. So just a second, there was a question about the refraction index. Okay, so it's the question from the same person. So what is the refraction index? It's the ratio between sinus of the incidence angle and the refraction angle. Okay, so whenever we have the border between two media, so the first media is air, and the second one is water. So let's say we have some light ray that comes under the angle to the normal. So this is called normal, and it's perpendicular to the surface or to the border between these two media. And we have this incidence light. Right. And we have the angle here, and this is the incidence angle. And after it falls here, some part of it will be reflected, and the other part will be refracted, which means changing the angle, it will still go almost the same direction as it was going before. And so we still have the normal here. And the angle here, let's call it beta. And this will be the refraction angle. So the ratio between sinus of beta to the sinus of alpha is the refraction index. And this index is used whenever we need to collect more rays that we could collect just a second. So I hope you understood this one. So I'm going to delete it. Okay, so let's say again that you have the source of light, and so light source. Then you have your sample. And then you have your objective. So let's say this light comes through your sample. So some kind of sample here. So the thing is, here we have air, here we have glass, here we have air, and this is also glass. Glass has higher refraction index than air, and it means it will the border between these two media will lead to the refraction. So we will have the bending of these rays, okay? So let's say it goes through it, and then when it leaves the media of glass, it's being refracted. So it goes the other direction that we lose. And it means that we do not collect all the information that comes through the sample. So let's say only the perpendicular rays will still go through and we can collect them. But the ones from the sides will be reflected, refracted, and we will lose this information. So what we could do, we could reduce the difference between the glass to air to glass. So if we reduce this refraction index that is here and here, this loss of information will not happen. So just imagine. So let's say this is the sample with the glass. So it's glass. And here we have the objective. And if we put oil here, that kind of covers both glass surfaces, and this oil refraction index is almost the same to the glass reflection index, then all the light rays that come, when they go through this limit or border of the media, they will not be refracted, they just go through and we collect more information. Instead of going like this ray will still continue its path and finally reach our eyes. To prevent the refraction problem, can we change the angle of coming light? Will it change the picture? If you change the angle, you will lose much more information because you will have a lot of scattering, a lot of refraction and reflection. That's why we use parallel, perpendicular to the surface beams. Only in dark microscopy, we intentionally illuminate it by the angle, but still we need a lot of this light to obtain the picture. And still if we have very different refraction indices of the media, we will still have the problem with the refraction. That's why we need to use oil. We can also use water. It's still big difference between water and glass, but it's lower than between glass and air. And the dark field microscopy. Okay, so we have two types, like bright field and dark field. So this is the same objective. And we have the samples too. And let's say they are the same cells. But in bright field, we have these parallel perpendicular light rays. And some of them, because of the structure of the cell, they are not being scattered. And scattering means whenever you hit something, you kind of bounce over it and change the direction, and sometimes drastically change the direction. And if you just go to the perpendicular place, you can go through this object, but most of the rays will be reflected, some with higher refraction, others lower. Reflection, not refraction. So in this case, some of the structures of the cell will not reflect the light at all. That's why every single ray that goes through it will pass. So let's say this guy goes like this. And the ones that come to, let's say, nucleus or mitochondria, they will kind of bounce of it, out of it. And this guy will also do that. This guy passes. So again, on the picture, you will see the same. So some of the light will go through, and we actually see this part of the image. I mean, the cut of these light rays. And if you think about it as the plane and look at it from the top, you will see something like this. And because you don't have light coming from this place, you will see darker. But generally, everything else will be bright. So generally bright. And this is called bright field microscopy. In the dark field, you intentionally try to catch the scattered light. So these guys which were bounced out. And to do that, you actually use the angle. So light beams, sorry, light beams come with the angle, and some of them go through. Some of them, when they find a place to bounce, they bounce. So, and others just pass through it. And whenever we kind of get the image, again, you have this plane. And if you look at it, you will only see the guys that are coming from something that they are scattered from. Okay, so you will see very bright this guy, and everything else will be dark. Of course, very little amount of light will come from cytoplasm, for example, Golgi apparatus, etc. and it will be very, very faint. That's why you will hardly see the other structures, only the ones that have scattered the light. And this is the difference between bright field and dark field microscopy. So here you intentionally scatter light and see the objects that are usually dark on bright field. And this actually increases the contrast, because whenever we have a lot of bright pixels, it means that a lot of artifacts and noise come with it. And whenever we have very little light that comes from the object, we can differentiate between the structures much more easily. So we will see mitochondria and some even smaller objects, and we can distinguish much more details than in bright field. And that's why this technique was actually developed. And the question about the active transport. Okay, so what is the active transportation? So we have the bilayer of lipids. So this is lipids. And this part and this part, they are hydrophilic. So which means they like water. This huge part is hydrophobic. Full of fatty acids. And not everything goes through both of these layers. And diffusion hardly happens. Also, it happens not only dependent on the concentration, but also on size. If you have a huge molecule, you will most probably not pass through it. So small size and concentration dependent. Also, it needs to go through both of the layers of hydrophilic and hydrophobic. So next we have facilitated diffusion. So we have a protein that, let's say, it can only give away to sodium, and only sodium ions can pass through it, not anything else, even water. And because of this concentration gradient, this sodium goes inside the cell. And finally, we have a protein that uses ATP to make it possible for some molecule go through it. So these two guys are passive transport, and this is active. Why it's active? Because it actively consumes energy to make this molecule go inside. One of fatty acids in tail of phospholipids is killed. The thing is, you can have many fatty acids that will be placed in this tail. They are usually shown like this. So one is like this and other is like that. It doesn't matter. Some of them might be just like this. Some might be both like this. The thing is, we have two fatty acids. So in glycerol, one of the carbons is occupied by phosphate group. And this is what is important. So 2 carbons are occupied by fatty acids, and 1 is occupied with phosphate. That is what is really important. This genus here means we have a double bond between these 2 carbons. That's why we have the change of direction of the chain of carbons. And it just means, this thing here, this articulation, it means that we have one fatty acid that is unsaturated.